Arts. I'm happy to say that my group are the hardest working people, and I participate to some extent. They let me participate to some extent. And so it's my pleasure to be back in the land of the felled animals on the roadside, the Happy Valley. Holy cow, there's a little overpopulation problem. I don't know what's going on with that, but to tell you about the sort of world of electrochemistry generally and protein electrochemistry in particular. For those of you that were here last time, I have changed things up somewhat, but not enormously because the field hasn't necessarily changed. Instead of 90 minutes, I have 45, so I'm going to try to step lively. And basically, putting this talk together was me throwing out slides, and so we'll see how it goes. So as I was trying to juggle this and the many other tasks to do, I thought of how am I going to be able to give this talk in 45 minutes, and so I looked across my table into the steely cold eyes of Casey, my pupper, and she said, well, just tell them what you know and what I like to know about. And so what are the things that we want to know about in the world of electrochemistry? And so just a quick show of hands, how many of you have collected a voltammogram of some kind? Yeah, that's pretty good. So a lot of you know a lot of the things I'm going to tell you about. In the world of protein electrochemistry, things do change somewhat, and those are some of the highlights that we want to try to hit on today. So with respect to keeping that in mind, yeah, this is going to happen. This is going to happen a fair bit. Because it's late in the day and you're tired and I want to take a nap. So this is what's going to happen right now. But the things that we might care about might be something that's, quote, simple, as measuring reduction potentials. What I would say is actually not that simple and is incredibly powerful, the type of thing that theory can't really calculate. Honestly, theory can maybe give you relative potentials, but not necessarily absolute potentials, I would argue, that are accurate. Accurately measuring reduction potentials that can depend upon so many things, as I'll try to illustrate in a couple of cases. Things like electron transfer kinetics, which would really be interfacial electron transfer kinetics, or perhaps modeling internal electron transfer kinetics. And then thinking about the coupled processes or binding processes, things that might involve proton-coupled electron transfer. So proton binding will be certainly something we want to touch upon. And of course, catalysis. People like to have electrocatalysts. These days, it is a growing problem, a growing field. New journals in catalysis science, which involve a lot of electrochemical detection, and sometimes that could involve an enzyme or not. And so, there are experimental tools that are associated with these experiments. A lot of them are effectively voltammetry of some kind. And so that might be just cyclic voltammetry. That might be cyclic voltammetry where we care about changing the scan rate, potentially to very fast timescales, or looking at things systematically as a function of time, and that time scale as a way of trying to modeling things, or using so-called square wave voltammetry as a tool for monitoring things with a temporal dimension I'll try to mention again today. And with respect to electrocatalysis, again, that means that we'll be thinking about potentially voltammetry in a different time regime, but also possibly amperometry, and I'll highlight that a little bit as well to try to understand how an enzyme is at work, and that idea of sort of the question that was asked about NMR. Here we very much want the enzymes to be at work at an electrode. And again, just from the standpoint of thinking about your particular metalloprotein of interest, if any of you don't study something that's redox active, I feel sorry for you because you're missing out on the beautiful world of electrochemically active things. And so just this idea of, for whatever kind of iron sulfur cluster in this case, or if we had a heme plot as well, or blue copper centers, that there are ranges of potentials, and we become very potentially obsessed over exactly what the potentials are of different clusters in different contexts and how these things evolve. And so this monitoring of redox potentials becomes something that we care a lot about. So from the standpoint of electrochemistry, really going back to that sort of idea of what is happening in electrochemical analysis, we need to have some type of electrode. And so this is, again, a textbook diagram, something where we can imagine having an idealized electrode, like a mercury hanging drop electrode or some other sort of hypothetical metallic electrode. And the idea is that what's happening at that electrode is a lot of physical chemistry. Whatever we're doing in terms of either applying a potential or driving the process in terms of the extruding of current, there needs to be a mitigation of species coming up to that electrode surface that involves considerations of an immediate layer and then an even secondary layer and potentially beyond to the diffusionable species that can bear charge, that might not bear charge, but might influence the type of analyses that we have to think about. And so that sort of beginning picture of what might be happening in electrochemical analysis of having an electrode that we're going to be doing something with in terms of a circuit, and then there are changes that are immediately happening at that electrode interface, this solid-liquid interface, is very much a part of the beginning point of just developing a voltammogram, of thinking about how is the right way of imagining what these type of electrochemical processes are about. So again, in a very simplistic Barden-Faulkner, which is kind of the electrochemistry Bible, way of thinking about a voltammetry experiment, we can have a linear sweep voltammetry experiment where, as a process of time, with some type of rate associated with that change, we're going to change a potential at a working electrode. And we can monitor the flow of current with respect to a different electrode, and monitor exactly what that potential is with respect to a different electrode. And the idea is that as that is occurring, if we imagine the monitoring those electrons that are passing at that bilayer interface in the form of a current, that is also going to occur as a function of time. And so the notion is, as soon as we start to ramp potential, that there's a charging up of this surface. This is surface interaction that's associated effectively with a capacitance. It's not just the capacitance, it's a capacitance that's also being modified by the scan rate, the time scale with which we're making that change. But that gives rise to that sort of curved onset of our temporal evolution of current. And so if we imagine that, again, in a cyclic voltammetry experiment, then we're going to sweep one way and we're going to sweep back again, and that would be one cycle. And then, of course, experimentally, you can do that many, many times, which might mean that you're capturing electrochemical information that's evolving as a function of time. But if we look at that sort of, again, time-based change of potential in terms of current, that means we have that charging up and that discharging process that occurs in a reversible sense. And this is what gives us our sort of football-shaped voltammogram that really is our background. And this is where, from the standpoint for those of you that have worked with anything that's biological, not using a metallic electrode, but a graphite electrode or a carbon-based electrode. And if I have any time at all, I might come back to that idea of what goes into making an electrode. Usually this capacitance is actually pretty large with respect to the type of signals that we will care about. And the shape, again, that sort of curvature is something that's unavoidable, but as well tells you something about the electrokinetics of the system, whether or not you have enough counter-ion that's present. And again, from the standpoint of if you're trying to detect something on a potential scale that might be associated with that regime or that regime, then you have a very curved background that's built into your experiment. And trying to mitigate that might be something that you can do by simply changing the scan range, but perhaps not, depending upon your particular experimental consideration. And so, again, thinking about this in the context of proteins, the idea is that we've got a little bit of signal on top of a lot of background. So a lot of time people are going to, it's a little signal, and so we want to capture that. It's dynamic, a great photo. And so, we're going to subtract away that background current, and that's frequently what people do. Sometimes they tell you that they're doing that, and sometimes they don't, and then I get angry. There's a lot that's about me getting angry. So if we have a diffusional voltammogram, then we could have a diffusional shaped voltammogram, the sort of classical duck-shaped voltammogram that's shown for you here, with the American convention of having positive things awkwardly shown on the left-hand side and negative things awkwardly shown on the right-hand side. But the idea is that for a simple redox process, whatever that might be, we can imagine there being a sort of diffusional process by which we can remove away that background capacitance. And there are rules. There are, again, equations that involve diffusion that are associated with that shape of the voltammogram. And a lot of what gives rise to that voltammetric shape really has to do with diffusion and not necessarily the interesting aspects of the electron transfer processes. And so we can make use of fixed laws for diffusion. These mean that effectively we always have to worry about what the diffusional coefficients are. And so again, in a time-dependent fashion, we can imagine if we step potential instantaneously and we can change the population of oxidized or reduced species, that those change both as a function of the temporal dimension, where that x-coordinate now means stepping away from the electrode, but also they change as a function of time. So if we gobble up the oxidized species that might exist, that changes as a function of time. And we populate the generation of these reduced species as a function of time. And so that notion then of that time dependence, diffusional dependence of things, let's say complicates things or makes it a sort of interesting possibility if you're working in this diffusional regime of having objects in solution that you're then interrogating electrochemically. Again, there are equations that tell us how this works if we engage in these processes such that we can actually solve for what the diffusional coefficient is in real time. And the Cottrell, that's the name associated with the Cottrell Scholar, that any of you that want to become young faculty want to become a Cottrell Scholar from Research Corporation, it's this Cottrell. Actually, Cottrell was a very prolific commercialized electrochemist, basically, in terms of the development of analytical tools that include electrochemical tools. And so with respect to that Cottrell equation, the notion is that this gives us a way of thinking about what will that current be in terms of a diffusional coefficient that will change as a function, and that will have an amount of current that is associated with the change of time as well. And so the idea being is that we have diffusion that's at work, and whether we use those unfortunate American conventions or instead the European conventions, which sensibly puts negative things on the left-hand side of our coordinate and our positive things on the right-hand side of the coordinate, and we think about oxidation processes as having positive current and reductive processes as having negative current, which just makes sense, we still have diffusion in that regime, which means that there is going to be a certain amount of peak separation that occurs and a certain degree of asymmetry that is at work with respect to that background signal that is addressable. But for sort of the things that I won't have time to talk about today, but there are tools, graphical tools, that tell us about exactly how the peak height, the magnitude of these signals, should change as a function of scan rate when we're in a diffusional regime. And that's summarized for you here in terms of both the peak separation and how the peak height, that maximal current, in either the oxidizing or reducing direction, should behave as a function of scan rate, and it should go as a square root of scan rate, where we still will have the diffusional coefficient that comes into play and will be related to, but is not directly proportional, linearly proportional to the number of electrons that are associated with that redox process. So that's sort of great, and it's great because it means that that's something that we can calculate. Again, there's a set of software tools for reconstructing diffusional voltammograms if you use DigiSim or COMSOL or different programs that are available. Most of them are not free, but that you could actually analytically analyze the wave shapes of diffusional species as you will. However, as the point that we'll get to is that we want to step away from this diffusional world into a world where things become immobilized. But before I get there, I just want to bring to your attention the fact that, again, this is the cyclic voltammetry experiment that we've talked about thus far. It's not the only tool in the game. Square wave voltammetry, or sometimes people will make use of differential pulse voltammetry, or other tools that are similar to that, are also routinely utilized. And so an example of that, the change of time with respect to potential is shown for you here, where in the case of a square wave voltammogram, we're going to step in this square-toothed pattern a sweep of potential that could be rising or could be decaying, depending upon what we use as the beginning point and end point of the potential. But now there's a lot going on instead of just a linear change of potential as a function of time. So we're both changing an amplitude, that becomes a parameter that we can alter, with the size of the jump with respect to that sort of general stepwise increase. And then as well, the amount of time that we hold that poising potential, that ramping potential, that jumping potential, is another variable that we can control, and that can be thought of as an actual amount of time, or frequently this is referred to as a frequency with which you are oscillating backward and forward in these jumping fashions. And the idea being is that with respect to square wave voltammetry, why would you do that when cyclic voltammetry is so nice? The simple notion is that in the idea of a square wave voltammetry experiment with a current that is displayed, and so here's just some data from a report from our lab with Squire Booker's lab studying an enzyme called BTRN, is that one really typically illustrates things in terms of a difference data set. You have both a forward jump and a backward jump current that emerges, and that if you subtract those two from one another, you arrive at something that, as a difference type of analysis, might eliminate more noise and give you an enhancement of signal to noise, and then that becomes a nice way of picking out things with respect to a curved background, which again can be subtracted and illustrated as such. And the idea being is that square wave voltammetry is great. I'd say it's increasingly being used routinely because it is more sensitive. And so for the type of things that people are interested in terms of protein electrochemistry, frequently in the cyclic voltammetry world, as we'll see, it gets hard in terms of what you're trying to pick out from that big background. And so square wave voltammetry can be a handy way around that. But at the same time, the problem with square wave voltammetry is that as it is a difference measurement, you could be blind to what's happening into one half of the scans if you don't actually plot that data, which few people do. And so comparing what is occurring in a CV and a square wave voltammetry is a smarter way to go. And so with respect to that, we can now start to move into actually collecting some voltammograms or showing some voltammograms. And so here's the case of cytochrome C2 from Rhodobacter. So this is just a sort of simple monoheme cytochrome C that gives rise to a very nice diffusional voltammogram. And the point that I want to try to make here, so with respect to thinking about this as a analytical chemistry problem, one of the nice things that's sort of alluded to about electrochemistry is its sensitivity. We can have devices that monitor small numbers of electrons, but if you go through the math of how this experiment, which is from Sola and Battistuzzi, it's this lovely paper, a series of papers from their lab studying monoheme cytochrome Cs of either bacterial or mitochondrial or eukaryotic origin. In this diffusional mode, basically you still need relatively large amounts of protein if you need to make a mg of protein to get a sufficiently high concentration, which in this case is not really that sensitive, but still 200 micromolar concentration in order to get these nice looking diffusional voltammograms. And so, we have turned, and people that are sort of trained in the Fraser-Armstrong inspired background of protein film electrochemistry, of immobilizing things, of taking proteins, absorbing them onto electrodes, dealing with the fact that the currents are going to be small, but give rise to a linear dependence of the scan rate in terms of the peak height. And the idea is that now we have an electrochemical area associated with things that are just absorbed right on the electrode surface, and that we basically have a very sensitive now reporter of looking at a small number of objects, relatively small number of objects, and that then gives us that sensitivity enhancement that we would like to have in our protein electrochemistry world. And so with respect to that idea of immobilization now, in principle, we should have no diffusion. That is a part of the equations that are describing the overall wave shape. So we could rapidly read out midpoint potential. And then the idea is that the peak height now will be dependent upon the number of electrons in a squared fashion. So if we actually have a multi-electron redox process, these should be more intense than they would be otherwise. And of course, there's still peak height and overall magnitude of the currents will be associated now with what we can think of as a surface concentration, a gamma term that we can imagine as being the electroactive surface coverage. That's not necessarily the number of molecules that are absorbed, but the number of molecules that are absorbed that are talking, if you will, with the electrode. And then the shape, the peak width, is associated again with the number of electrons that are associated with that redox process. And so, there are these aspects of analyzing the data analysis that ultimately tells us that we are looking at the number of, we're counting the number of electrons that are associated with species that are directly absorbed onto an electrode. But in reality, so this is the bit of the gut check here, is that frequently we do see some peak separation between anything that we've looked at electrochemically, and I think anyone else in the protein electrochemistry world typically observes some small degree, smaller than what should be present for diffusion, but unlikely, most likely this is just due to, relatively speaking, slow interfacial electron transfer kinetics, that there's some dispersion that exists as well upon the electrode surface. And then as well, this notion that currents are not necessarily big if we needed to have a lot of material absorbed on the electrode. And then at the same time, the sort of sensitivity that we get back usually is most helpful because we know that there are specific equations that govern these shapes so that we can deconvolve complicated voltammograms into multiple components. And we can, as well, as I'll show you momentarily, understand that, of course, if we have a sense of cooperativity in, say, a flavoprotein or something that might engage in 2-electron transfer chemistry, we can winkle that out from the voltammograms. So, why is the current so small in these cases? And basically, it's a steric argument, again, that these are big biomolecules. And they are big biomolecules occupying a large chunk of your space on your electrode. Ultimately, we need to have things that are interacting with some type of electrode material. And a kind of cartoon depiction, which, as I frequently say, is a nice cartoon, but we know it's exactly wrong. That the electrodes themselves are not smooth objects. Usually they are very rough because proteins themselves are rough. We can't necessarily orient proteins very nicely, although if I have a little time at the end, I'll try to tell you about some tricks that people are using to orient things more specifically. But even then, when people do orient things quite frequently, they just covalently enforce a population upon the electrode surface, and you end up with more dispersion than you might have otherwise. But the key idea, of course, is that there's going to be some population within this that is electrochemically active, that is capable of fast electron transfer, fast enough that we see actual steady-state voltammetry that results in either a voltammogram, whether it's a cyclic voltammogram, or a square-wave electrochemistry experiment, or in the case of an enzyme turning over in some type of amperometry experiment. And so we can see that again in another case study. So this is a scenario of a flavocytochrome, so where we have a buried flavin that is the active site of this fumarate reductase, and then that's wired to the outside world to a series of hemes in this Schwanella FCC3, as it's called. And so that's a scenario where in the absence of substrate, the raw voltammograms look as such, and it's subtracted in a way that what's referred to as the non-Faradaic component of the current, just the background charging up and discharging of that graphite electrode, is very nicely symmetrical, but not perfectly symmetrical, but still quite symmetrical components that are associated with the FCC3. And then by varying pH in this case, the lab of Armstrong was able to deconvolve this into a series of features that are associated with the 4 heme cofactors, and again, this narrow and more intense flavin peak, that sort of idea of the signal. And so this is where, the problem is that if things are too close in potential, can you really tell them apart? Here you were able to do, you were able to do that well here because one of these things really stand out with respect to the others. And so you need to have enough separation of potential in order to be able to tell things apart from one another, and that some things need to be taller than one another as well if they're really going to be superimposed in that way. So it's convenient to have big features and small features, sort of the bottom line, or at least to have them very well separated in terms of potential. In terms of this idea of cooperativity, again, when we think about electrocatalysis, a lot of the electrochemically interesting reactions that one wants to do are not one-electron reactions. And so one of the sort of pressing questions in the area of bioinorganic chemistry is how do you do multi-electron redox catalysis using cofactors that only handle 1 electron at a time? So understanding the nature of the redox processes associated with either active sites or with cofactors, be they flavins or other cofactors, that are capable of multi-electron chemistry is really an interesting question. And so there are ways, again, of modeling how that should behave in terms of direct electrochemistry. And so if we think about a system that could cycle through 2 different redox couples, effectively going from an oxidized version to an intermediate version, that intermediate version then being reduced all the way down to a reduced version, the question is basically how separated are those individual 1-electron potentials? If they are truly separated, then we have a scenario like in the case of the green line shown here in this review from Christophe Leger, indicating that we should see these as 2 separate 1-electron features. And as those formal separations of individual 1-electron redox couples become closer and closer, there's eventually a point where they're coincident and still behave as the sum of 2 1-electron features. But as they become crossed, then you see that sense of cooperativity that emerges and the increase of peak height. And so this has evolved to a equation that is frequently utilized, the so-called Laveron equation that people will refer to with respect to this sense of cooperativity, generating a parameter that's referred to as N-ap, or this apparent number of electrons, and then this is the version of that equation that frequently gets utilized in the protein electrochemistry literature. And I want to highlight that because while people get confused as to what does N mean? What does N-ap mean, this apparent N value? And the fact of the matter is that it doesn't really have a physical meaning. It's a phenomenological parameter. It is associated with the number of electrons that are associated with that. If it is greater than 1, that indicates a sense of cooperativity, but 1.2 isn't necessarily better than 1.4 with respect to that in a quantitative sense of a physical parameter that means something. The better way to treat that formally is to think of the inversion, the potential inversion of those individual redox couples. And so that's just a sidebar point as well. But with respect to other problems, again, whether we have flavins or in the case of this work from our lab on thyrodoxins, this is a scenario where, again, we're picking up very small features in terms of this background of the blank electrode response, but they clearly have a sharpness that is associated, not necessarily in terms of peak height, but in terms of the width parameter, that delta parameter that tells us that they're behaving somewhere in between a 1-electron couple and a 2-electron couple. And in fact, this is a case as well where we were able to show that for a variety of these different thyrodoxin proteins, basically that really depends upon temperature, and that the cooler things are, which lends itself to mechanical stability of the protein-electrode interaction, so it will stay around for more scans. If we heat that up, it actually behaves in an opposite fashion as the theory would predict, or tell us that it should slightly increase. Instead, if anything, it's heading more and more toward the 2-electron limit of what would be theoretically conceived for a truly cooperative 2-electron process. And then as well, we can have sort of simple things that give rise to more complicated features. And one example of that is, not that there's a textbook of protein electrochemistry, but one of the best case studies of protein electrochemistry in the literature is the Azotobacter vanlandii ferredoxin 1. So this is a ferredoxin that has, as you can see, 2 iron sulfur clusters associated with it. One of them is a 3-iron 4-sulfur cluster. And that 3-iron 4-sulfur cluster has a very unusual, it is presumed, 2-electron redox couple, giving rise to this very low potential, very sharp, again, 4 times as intense feature as would be observed with respect to the other normal, if you will, redox potentials. And so whether or not that is operative in terms of Azotobacter finlandii ferredoxin 1's functional role is to be determined still. But the idea being is that you can have processes that you might understand the bits and pieces thereof in terms of the type of cluster chemistry that might have, but until you do the electrochemistry, you don't really know what those redox properties can hold. So what about binding? Oh yeah, binding. Yeah, we like to bind things, particularly if they involve sticks. Thanks, Casey. So the idea being is that if we want to look at binding phenomena, and typically one of the most typical things that we want to consider in that regard is proton binding, thinking about proton-coupled electron transfer. One of the things that is most often invoked but most rarely shown, that means you have to measure a Pourbaix diagram. That's sort of the bottom line from my perspective. Is that if you want to demonstrate that something is proton-coupled electron transfer, you have to show that proton goes with the electron in a way that is in fact coupled. And so that basically means that we would think of a redox process as existing in the absence of a proton or potentially in the presence of a proton in a coupled fashion such that there could be protonation events for the oxidized form or a protonated event for the reduced form, such that you can collect the redox potential, measure the redox potential as a function of pH, and then that gives you a sense of that notion of coupling, and if they behave in an Nernstian fashion in a so-called Pourbaix diagram in a one proton, one proton, one electron coupled fashion, or with some other stoichiometry of electrons to protons. And so again, a few examples of this has been measured in our group for the protein I alluded to before, BTRN, which is an 8-O-met radical enzyme. It has this very curious active site with respect to other 8-O-met radical enzymes and frankly other iron sulfur clusters where there's a very clear Pourbaix diagram, proton dependence of one electron, one proton for the 8-O-met radical cluster versus a secondary cluster, the so-called auxiliary cluster, which sort of has a generally weak dependence, which is what you typically observe for other iron sulfur clusters in either ferredoxins or most other proteins that have been examined in the same way, compared to other, iron sulfur cluster proteins that, generally speaking, whether they be high pips or ferredoxins, do not show a significant proton dependence that can be directly linked to proton-coupled electron transfer. The examples that deviate from that all require histidine ligation, where it's presumed that the proton binding event is occurring at the imidazole nitrogens, the auxiliary, either the delta or epsilon, the other nitrogen that is not directly ligating the iron center. And so one example of that is in the case of risky type of proteins, studied by Judy Hurst. From our lab, we have studied, again, a protein called mitonete, which has this unusual set of 3 cysteines and a single histidine residue, but clearly has some very complicated proton-coupled electron transfer stuff going on with respect to multiple pKa's that are at work. And we've examined this exhaustively in terms of making mutations to try to understand the nature of exactly what is giving rise to the proton coupling and where do the protons go, as well as the electrons. And then in this way, we can think of protein electrochemistry, and particularly voltammetry, as being a tool for detecting things. And so if we have a set, as we increasingly do, of having not one cofactor but multiple cofactors, you could imagine that just being able to observe the redox potentials associated with these individual species tells you that they are present. And so this ability of electrochemistry to have a time dependence associated with that has been used in our hands in the case, again, in another Booker Lab collaboration with the proteins MIA-B and RIMO. Their functions, for our purposes, don't really matter, but simply that they have 2 clusters that are very close in potential across members of this family. And we've used square wave voltammetry again to try to assign which potential is which, albeit however close they may be. But most importantly, for this idea of protein electrochemistry as a detection tool, we found that in the case of MIA-B and RIMO shows the same effect, that as we add 8-O-met, this sort of general sense of close potentials changes as a function of time. And so again, this is a sort of slow time scale, but these are slow enzymes. And the idea being is if we look at the square wave data or in that baseline subtracted data, there clearly are changes that are occurring in the voltammograms as a function of time. And so square wave data you can collect on the time scale of seconds, so you could, or longer time scales. So this notion of if it has a different redox potential, if the clusters are changing, if ligand binding is happening, you can detect that electrochemically and pair that with spectroscopy. All right, so 15 minutes left, I think, and we'll talk about catalysis. So in the case of electrochemical experiments that one wants to couple to the conversion of substrates to products, One additional component that we have to think about is how we control the concentration of substrates and the removal of products at the electrode surface. Again, going back to that very fundamental idea of the electrode surface where we need to be able to get stuff up to it if those things are going to be diffusional. In the case of this type of analysis, we want to have our catalyst, in this case a protein, but even if it's a small molecule catalyst that is adsorbed, we still need to be able to sweep up substrate molecules and release product molecules rapidly. So that requires some type of rotating electrode or some other type of electrode configuration that has flux in it, that has a sense of moving substrate molecules up to the electrode surface and carrying away the product molecules in real time. And so the conventional tool that's utilized is a so-called rotating disk electrode, where again, the analytical equations associated with diffusion have been solved for that problem, so we just know how to handle those currents very well. And the idea being is that in contrast to a non-catalytic regime, now if we're in a catalytic regime with no diffusional control, as we're sweeping our way, we should see the transformation of our non-turnover signals potentially into a sigmoidal wave shape where we have an actual limiting current if the enzyme, if the catalyst is moving as fast as it can. And the idea is that the maximum amount of limiting current that we could observe in that sense is thinking about the enzyme turning over as fast as it can if that happens in a potential independent fashion, which is a notion that I'll show you in a little bit isn't really the way things work in most cases, quite honestly. But the notion then of how this electrocatalytic behavior is tied to a sigmoidal point, a midpoint potential, is very useful information that we would argue in terms of mechanism. Whether or not this is in this case just a 1/2 reaction for an enzyme or a catalyst that might be reversible, so perhaps this would actually cleanly go through the redox process associated with the formal thermodynamic redox potential of a reversible process as well. So for those of you that are more enzymology or enzymologically driven, basically to think about these cyclic voltammetry experiments, ultimately what we're trying to do is assess, what is the velocity, what is the steady state turnover properties of the system, where now we're instead of having some type of externally determined turnover rate through whatever assay you might use, we're going to make use of that limiting current as a function of time. At the same time, there's going to be complications again, insofar as we have to think about exactly whether or not the absolute magnitude of the limiting current matters, because that will be in part determined by the surface coverage. And so if we don't have very much surface coverage, then we might not actually have that much catalytic current. But at the same time, the amount of catalytic current should depend upon how fast the enzyme's turning over, ultimately. We'll see that there are cases of film loss, the ability to turn on and turn off an enzyme on the basis of what oxidation states are populated, the potential complications of mass transport. There's a lot of questions that have to be asked anytime that you want to examine something in terms of its electrocatalytic behavior. But at the same time, this sort of cyclic voltammetry experiment, where we sweep potential, will be complementary to amperometry, where we just hold potential and then can watch the instantaneous evolution of steady-state current as we change a parameter like the incubation of an inhibitor, and I'll show you an example of that. So if we go back to the sort of theory in terms of what's happening in electrochemical analysis, one can imagine about changing the time scale of the experiment, literally by speeding up or slowing down how rapidly we cycle through the range of potential that we visit, or by imagining in a theoretical concept of being able to say, well, what if I had a catalyst that was a very slow catalyst and I could make it faster and faster and faster and faster? And so the idea is this set of voltammograms illustrates that transformation of if we maybe take that latter idea of saying, if I have something that is frankly not really a catalyst, and then start to turn on catalytic activity, the wave shapes should transform into something that looks like just a redox process that then takes on that sigmoidal shape. And you'll see that in the case of these voltammograms, this goes as well from the standpoint of current, these deconstructive voltammograms, that goes into a sense of current that grows and grows and grows and grows until the enzyme, in this case, the catalyst is actually quite fast, which we would think of in terms of Kcat or K2, in terms of the kinetic model that's used. And that same idea can be visited by manipulating the time scale of the experiment. And so this is where, in the standpoint of how one does electrocatalytic work, frequently we're using scan rates, if we're doing cyclic voltammetry, that are slow, because we want to maximize the lambda factor. We want to maximize the amount of current that we observe. And so we let the enzyme have a chance, if you will, to do its job, to be at work. But at the same time, if we jack up the scan rate, if we can literally try to outrun catalysis, it's like going from this type of plot to this type of plot by showing that we are outstripping the rate by which the enzyme can turn over by moving electrons into and out of a given redox process that may be at work. Again, the ability to visit a sense of mechanism insofar as identifying the redox process that correlates with our ability to see some type of catalytic electrochemistry. All right, so the sort of key point with respect to that is that, a lot of the shape of a catalytic voltammogram, whether it's an electrocatalyst, that's a small molecule, a heterogeneous thing, or an enzyme, will depend upon the nature of KCAT and the nature of the catalytic model that's at work for how it's going to transform. And so the idea, again, is that we could have something that's very active and hard to get a high-resolution picture of, or something that's really slow and easy to get a high-resolution picture of from the standpoint of the voltammetry. And so that means that when you look at the literature of protein electrochemistry of enzymes in motion, or catalytic voltammetry, that the shapes of voltammograms are highly different. And so again, this is from a series of reviews from Christophe Leger, collecting data from various systems. Some of us ours, some of us his, some of us from the other Armstrong lab. But it's showing that you can have the nice cases of limiting current, potentially for reversible enzymes. You can have cases where there's a diminishment of current for this reductive enzyme, where there's this business of reductive deactivation that then is recovered as a function of potential. So seemingly omic behavior is found in many of the cases of hydrogenases. In cases like nitrate reductase, cytochrome C nitrate reductase, nitrite reductase, that show these very complicated voltammograms where there's minima and maxima that are recovered as you sweep to even yet lower potentials. So developing a sense of what that mechanistically means is challenging because you need to work your way through this sort of list, this checkbox of what exactly is going on in terms of the types of traits of chemical behavior that we can know things about. What might be giving rise to these different phenomena, and what are the type of information or the type of questions that we can ask. And so some of that is illustrated for you here, and I'm going to take you through a couple examples again to try to show some of these differences that might be at work. So in the case of reversible enzymes, not all enzymes are reversible, but from the standpoint of how we can imagine that playing out in terms of electrochemistry, we think about something that can interconvert fumarate and succinate. This is the case of SDH, that you basically can have a perfectly reversible voltammogram that cleanly passes through the thermodynamic potential visited for that reversible redox couple, where there's a positive current associated with oxidation, a reductive current associated with reduction. And the idea is that depending upon the Km values of those two substrates for this particular enzyme, you might have to max exactly how to balance those amounts of individual current. And that is a phenomenon that you're not going to see for all enzymes, because not all enzymes are going to be reversible in that way. But the general sense is that one can make use of those limiting current values, whether in the raw voltammograms or in the background subtractive voltammograms, as a means of utilizing the limiting current as our indicator of Michaelis-Menten type of kinetics, of a steady state turnover value, and then use that to recapitulate ultimately a Km value that can be determined for a given enzyme. So that can be done in terms of those cyclic voltammetry experiments, or again, here's a scenario of using chronoamperometry, where one is poisoning at a potential associated with a catalysis. Then, as in this case with NAPAB, a nitrate reductase, if you titrate in nitrate, you are seeing these evolutions of increasingly negative current that eventually get saturated, because eventually you do saturate the enzyme through that addition. So it's in some ways, an incredibly fast way of determining a Km value for something which now has a negative appearance to it, because we've evolved those negative values of current. We've used chronoamperometry in much of the hydrogenase world as well for trying to understand different aspects of how hydrogenases of various kinds turn over. And so this is just one quick example of saying if we vary the poise, so we're showing these blue and red and green traces, but varying the poise with which we are poisoning the electrode and doing the exact same experiment over and over again. This work from my former postdoc Pierre Cicaldi, but of looking at either hydrogen oxidation or proton reduction in the presence of CO. The sort of question of asking, how does CO behave as an inhibitor, where I'm not going to take you through the entire set of slides, but the idea being that there's a mechanistic query about how CO behaves as an inhibitor, which states of hydrogenase are really the ones that are most important in terms of CO acting as an inhibitor in the case of these iron-iron hydrogenases. And so by visiting these different potentials, we're able to actually fit the chronoamperometric data to a very simple model under all cases and demonstrate that this HDCR enzyme is fully reversible. So the notion, again, the sort of point that you want to take home from this is that by analyzing the actual data, fitting it to a kinetic model, you can determine things about mechanism in the same way that you could from other mechanistic type of studies. That all requires that you have the system under control where you know that the currents are telling you about the enzyme or the catalyst that's at work. And so that idea of mass transport is actually a potentially important component for a given problem. And again, is something that not necessarily is evaluated systematically in every case. But the idea being that if one has, in this case, one of the first nickel iron hydrogenases that was examined electrochemically and worked from the Armstrong lab, it's demonstrated that basically you need to spin that electrode, that rotating disk electrode, faster and faster and faster to actually arrive at a limiting current that does not have diffusion as a component. In this case, that diffusion is just the diffusion of substrate, that the enzyme is so fast that it is going to gobble up all the substrate that's possible, such that you can keep going about to the point at which the mechanics of your rotator give out, or you start to get very worried about having a very highly, a lot of spinning velocity in a closed glove box. What happens when something breaks inside a box? It's not good. That's what happens. But the idea is that could be increasing entirely, and that is addressed in the Kutecki-Levitch type of analysis, which basically means that you want to extrapolate back to a point of potentially infinite rotation rate to get at what the genuine limiting current should be. So then, that's one type of complication. This notion of having maxima and minima associated with redox processes is another type of complication that can emerge. In the case of nitrate reductases, this has been fit to scenarios where it's thought that different manifolds of reactivity at an active site can participate in catalysis, but some of them have a better affinity for substrate than others. And this has not been actually fit in terms of directly modeling the data specifically, but has at least been conceived to be effectively understood in terms of preferential binding to intermediate oxidation states of the lipidopter and active site. And then one of these additional components of the hydrogenase world that's been widely utilized in terms of electrochemistry is this notion of activation and deactivation processes that can occur at an electrode. And the idea being that if you have an active site that might have states which are sensitive to redox poise, and some of those states might be more or less reactive, then you can see these diminishments of activity. And so in the case of this particular nickel iron hydrogenase, this is the greatest extent of inactivation that occurs that then is reversed upon revisiting the lower potentials. And then the sort of final little example that I'll tell you a little bit about is just this idea of we try to imagine in the world of catalytic voltammetry, whether enzymes or catalysts, that we're learning about the active site. That's the sort of goal typically, is to say that we're going to understand active site chemistry. But there are times where, frankly, processes are mediated processes, that the redox process that is telling you about information about the catalysis is genuinely associated with a relaying device. And that device, in this case of a sulfite oxidase, is a heme-based center that acts as an electrochemical relay to the electrode. And that's understood very well in terms of structure, and I don't have an actual X-ray crystal structure version, though these are available now, to show you. But the notion is that there's a domain that contains the molybdopter and active site, and a domain that contains this relaying heme center. And when one examines the electrochemical response, basically you see just the heme domain. You see a redox potential that's associated with the B heme domain that exists. And then when you examine its electrocatalytic oxidative behavior, oxidizing sulfite, you see a nice little small catalytic wave, but it's centered at exactly the potential associated with that mediator. And then by looking at its Pourbaix diagram, which doesn't have much of a Pourbaix diagram, but a very minimal dependence, and that's exactly what you observe for just the free heme cofactor itself, or I shouldn't say free heme, but the heme domain itself. So the notion being that really what we're examining in this case, the initial report when I was a postdoc, which is this idea that phenomenologically it seemed that what's happening is that the molybdopterin domain gets too far away, that we can't have direct electron transfer to it, and instead what we're really learning about is the relaying process of one domain handing off an electron to picking up an electron, I should say, from the molybdopterin domain in due course. And this type of phenomenology has been observed not just for internal events within proteins, but also between proteins. And so cytochrome C oxidase will turn over in the presence of its cytochrome C, where that is observed as a relaying event associated with the cytochrome C interacting with the oxidase. The Schwanella enzymes that are associated with long-range electron transfer will hand off electrons to one another only in certain pairs. And recently as well, we've addressed this with respect to the oxoacid ferredoxin oxidoreductase family, where we require the ferredoxin to act, the right ferredoxin to act as a relaying intermediary to hand off electrons to be able to reduce CO2 appropriately. And so I hope at the end, and I think I'm coming in on time, the notion is that we're seeing a bunch of different properties associated with protein electrochemistry. And hopefully now by the end of these 45 minutes, you have some sense of the challenges that are associated with examining proteins at electrodes, some of the opportunities that exist as well. So just the last thing I want to do is to thank my group. They are the hardest working people I know. And today, I should say not today, but tomorrow and for the next few days, you may have the privilege of working with Rhea, Lindsay, Kim, Madeline, and Sheila, who will be doing the training sessions with you. And so if you can't find me for beer and questions, you can certainly ask some questions as well during your tutorial time. And with that, if we have a moment, I'll be happy to take any questions. If not, I'll hand things over to Hannah.